So what are these multifunctional mixed selectivity neurons? Well, again, back in the 20th century, we thought that neurons detected single things like edges, or a combination of a few things that that corresponded to like one percept. So a few neurons might combine color and form and motion to percept a rolling ball. But in conducting these studies, we found these mixed selectivity neurons that they were selected for complex combinations with no obvious relationship. If you train the animal in different tasks and study these neurons at different times, these neurons will seem to do different things, different functions in different contexts. And they seem to have no obvious relationship to one another. And they were multifunctional. So why are these mixed selectivity neurons so important for this top-down, high-level information the animal is learning? My colleagues Stefano Fusi, Mattia Regata, and Melissa Warden, they showed they did computational using data from Melissa. Stefano and Mattia did computational modeling that showed why these mixed selectivity neurons are so important. They add horsepower to the brain. They create a high-dimensional representational space where more complex representations can be represented, and that allows more complex computation. And it increases the storage capacity of the brain by distributing information across many neurons. If you build these toy models and every neuron does one function, the models quickly saturate and they can't learn more than a few tasks. But you start adding in these mixed selectivity neurons and you get to this number of about 30 to 40%, and the model goes capacity unlimited. And mixed selectivity allows faster learning and flexibility. Because all this information is intermingling, this wide variety of information is intermingling in the same population of neurons, things can be put together really quickly because the brain has all the information on the fingertips. And for the same reason, you can get flexible behavior because representations can be formed and recombined on the fly. Now, but this idea, and at first, again, this idea of mixed selectivity was met with a little bit of skepticism. We were accused of turning the cortex into a bowl of porridge and stuff like that. But, eventually now mixed selectivity is pretty much commonly accepted. I'm seeing new papers all the time about mixed selectivity here and there. Good thing. But this led to a problem with the old paradigm. According to the clockwork paradigm, anatomy is destiny. Neurons combine signals because they are wired together. So the idea was that every perception, every thought, every action, everything your brain does has a unique anatomical network called an ensemble. I've illustrated this here. Here's this network for thought blue, we'll call it. Another network for thought red. Separate networks, separate ensembles, separate bits of information, separate percepts, separate memories. But mixed selectivity means that neurons aren't members of one network. They're members of multiple networks. So not this, but more like this. Now I've combined our red and blue network into a single network that has anatomically overlapping parts. So we have our red neurons and our blue neurons, but they're connected. They share these multifunctional neurons that do both. They're part of both networks. Okay, so this is what mixed selectivity leads us to conclude. But if anatomy is destiny, how does this work? If neurons work by combining their signals, why don't all the networks activate one another and create a jumble of thoughts? I try to activate the red network here, and the red neurons activate, but then the activity runs to the mixed selectivity neurons, and then to the blue network, and now you have both networks activated, both ensembles, and your thoughts are a mush. So how does your brain deal with this? Well, one solution that many of us have come to, the solution is brainwaves. Your brain oscillates. Your brain is a highly oscillating machine. It's the most obvious signal you can record from the brain. It oscillates anywhere from 1 time a second to 4 times a second, 13, 30, up to 100 times a second or more. And what brainwaves are, they're an emergent property. They're the synchronized activity of millions of neurons, a large-scale organization of neural activity. So here's a crowd doing the wave. You can see the wave moving from left to right. Organization of large numbers of elements. But in the past, when we had this piecemeal view of the brain, brainwaves were often dismissed as being like the humming of a car engine. Humming doesn't make the engine run, it just reflects the engine running. But I submit to you that attitude was a byproduct of our thinking about the brain in pieces. Because if you think about it, if a stadium crowd, such as you see here, can self-organize using a few simple rules, like stand up and the person on my left stands up, imagine what your brain can do with that principle. So for executive control, you need this kind of large-scale organization for top-down executive control. You have to control whole sections of the brain so they're all on the same page together, coordinated and directed towards the task at hand. How are you going to do that on the individual neuron level? You're going to have a gazillion control gates on a gazillion synapses? You can't. That's possibly complex. But if you control things on this scale by these brain waves, where you control sections of coordinated sections of neurons, now you're at a scale that's useful for executive control. And also there's something called representational drift. Neurons are fickle. They come and go. If you record from 100 neurons or 1 million neurons and do 100 identical trials or 1 million identical trials, do the same thing over and over again, you will get 100 or 1 million different patterns of neural activity. The brain is kind of like the world's largest orchestra. The song plays on, but individual players are constantly coming and going. They're arriving, playing, and then they're leaving. That's how neurons work in your brain. There's a large amount of representational drift. But at this higher level, in this case, here's a study from Dimitris Panosis, where if you look at the level of, like now we're looking at the level of the electrical fields, the neuroelectrical fields that the neural activity in the brain creates. And he found that you could read the contents of working memory from electrical fields near the brain. But here's the trick, or here's the good part. There's little or no representational drift. It is a solid representational information channel. Now I submit to you, doesn't that sound like a useful level for organizing brain activity? If you want to get engaged in top-down control, pay attention, select certain things for processing, thinking about, engage in certain actions, connect things together. You got to do things at this level of organization where you're connecting millions of neurons together in a organized, coherent way. I don't see how you could do it by organizing things on the level of individual neurons. And isn't it easier when you get to this higher level, there's less and less representational drift? Isn't it easier to control a stable representation? I mean, if you want to conduct the orchestra, individual players are coming and going. There's no use to instructing individual players. You're instructing the sections of the orchestra, right? And that's the whole thing about this higher level, is that individual neurons come and go. The players of the orchestra are going and leaving and coming back, but the song plays on. The function is the song. So somehow there's a higher level of organization that has a stable representation that all these fickle players are contributing to. So the way to think about it, we think, is networks formed by synchronizing their oscillatory activity, their brainwaves. Think of them as two overlapping networks of neurons, shown here on the left, that are doing separate waves, separate crowd waves. The idea is that neurons that hum together temporarily wire together. And what endows the brain is cognitive flexibility. It takes a long time to make new connections in your brain or change connections. But with this kind of format where neurons that hum together, wire together, network can be formed, changed, all without actually rewiring your brain, but just by changing the resonance patterns of the brain waves. So you have resonance patterns stay stable until you flip them to a new format, then they flip to a newer format and they're stable there. So you have the combined stability when you need it and flexibility when you need it. That's what these patterns of brainwave resonance buy us. So the way to think about it is brain anatomy is not destiny, it's possibility. Brain anatomy is like the road and highway system. It just says where traffic could go. Your thoughts are where traffic actually does go moment to moment on the streets and roads that make up this infrastructure. So we think the brainwave, these brainwave patterns direct the traffic. They determine where traffic goes from moment to moment. Is there evidence for this? Yes, a lot. And here's just one example. Tim Bushman was in the lab. He trained animals to switch back and forth between two behavioral rules. They were looking at take that distraction out of the way for a moment. They were looking at either a vertical or horizontal bar, and the bar was red or it was blue. And you cue the animal, either pay attention to color or pay attention to orientation. You do that randomly from trial to trial. So the monkey's switching back and forth between two rules. And what this shows is neural activity in the lateral prefrontal cortex. This would be right here on the side of my head, about right here, above the animal's forehead, behind the animal's forehead, a little bit above its eye. That's posterior, that's anterior. This is the principal sulcus that runs down the center of the lateral prefrontal cortex. And each of these circles is a different recording site where we have one of our microelectrodes. The colored lines show which pairs of recording sites showed an increase in their LFP synchrony, their brainwave synchrony, if you will, when the animal was following the color rule versus the orientation rule. So you get different patterns of brainwave synchrony for one rule versus the other. They're overlapping, as you'd expect from a system with a lot of mixed selectivity, but you get unique patterns, unique ensembles, if you will, from these synchronized patterns of LFP activity. And we looked across all frequencies, and we found that these effects were mainly in the beta band, whose relevance I'll explain in just a moment. And that moment is now. So we also found we're finding that different brainwave frequencies carry different types of information, kind of like different FM radio stations. So don't make the mistake. All these frequency bands, like theta being 4 to 8 times a second, then alpha, then beta, on up. These were labels and designations someone made 150 years ago in a very arbitrary way. So there's not, beta is not just beta. The actual biology, leaks across these boundaries. And it's good. I think the function is going to be more like differences in frequencies rather than absolute frequencies. So don't get caught up in the labels too much. But what we found is that high frequencies in the gamma range, they're the signals that they're the brain waves that feed forward sensory signals from your sensory system in the back of the brain forward in your brain to your executive prefrontal cortex. So all this spiking that's carrying the information about what you're seeing, what you're hearing, etc., is all riding on these gamma waves. And then we found that the feedback signals that go from the executive prefrontal cortex back and modulate the rest of the cortex, they're carried by low frequency waves. They carry the top-down executive signals that regulate cortical processing. And this makes sense because it's easier to control large populations of neurons if you're doing it with lower frequencies and higher frequencies. And in fact, we believe that the imbalance between the sensory information is flooding into your brain, and your brain's got to regulate it because too much you can't process everything. So too much sensory information is bad. And that's what we think these low frequency oscillations are doing. And we think the imbalance between low and high could explain things like ADHD or even the sensory overload seen in autism. We think it's because the low frequencies are too weak, and then the high frequency sensory signals overwhelm the brain. So we're working on interventions to rebalance them.